Thanks, Barry. So it's been incredible for me and my student Danielle to be here. It's a real pleasure. We're microbiologists, so we're learning a huge amount at the conference, so thanks for everybody's comments and your patience with us. So the project is called ABATE, and the idea is that we would like to find bacteriophages that can kill the agent that causes American foul brood, Pennibacillus larvae. So as beekeepers, you know a lot about Pennibacillus larvae, and we've heard a lot about the problems that we have with Pennibacillus larvae here in New Zealand, a lot during the meeting. So I'm not going to belabor this point, but for me, the interesting thing about Pennibacillus larvae is that this is a bacterium, and that's key for us. So bacteria live in environments where they have a complex number of other environmental features that they're dealing with. These include things like protozoa and paramecia and different types of predators. This includes other species of bacteria that they're constantly competing with. We could consider the types of other cells that they encounter in the immune system of something like a bee or an insect to fall into this category, things that are kind of gobbling them up. They also deal with antibiotics, essentially, that are being produced naturally by other types of microbes in the soil around them. And these are the sorts of environmental features that we are kind of accustomed to thinking about. But one of the other things that all bacteria deal with in their environment is their viral parasites. And these are bacteriophages, or phages for short. So a bacteriophage might look something like this. So it's a small protein-based little entity that's basically just protein, and it has some kind of genetic material inside of its head. It doesn't swim. It literally just floats around in the environment, bumping into things. So what that might look like is the following. So a bacteriophage is kind of wandering around in its environment, and when it runs into its particular bacterial host, which can be very specific, and that becomes important, it recognizes the outside of that bacterium and is able to inject its DNA into that bacterial cell. At that time, it injects its DNA and it takes over the machinery of the cell, and in as little as 20 minutes, a single bacteriophage can create 100 copies of itself and then burst out of the bacterial cell, destroying that cell. So this not only kills the bacterium of interest, but it also amplifies the number of infectious particles right at the site of the bacterium. One of the things that is interesting about this is very much like antibiotics that might be dropped onto a filter paper, as in this slide. So here there are various antibiotics that have been put on filter papers on a bacterial lawn. Bacteriophages do exactly the same thing. You can drip bacteriophages onto bacteria and see complete lysis of the bacterial lawn. So these are incredibly efficient killers of bacteria. And in fact, for a very long time, close to 100 years, bacteriophages have been used as therapy and medicine in places like Georgia and Russia and Poland. And even today, bacteriophages are being used as life-saving treatments in the United States when patients are facing antibiotic-resistant superbugs. So for us, they're incredibly fascinating, very simple little entities, and we're very interested in how they can be used in the bee industry. The advantages of using bacteriophages to kill a bacterium that we're not interested in having around are, first of all, if we look across the numbers of these entities on the planet, there are 10 times as many bacteriophages on the planet as there are bacteria. So for every single bacterial cell, there are 10 bacteriophages gunning for it. They are highly specific to the pathogen. So if a bacteriophage is used as a therapeutic agent, it only kills the specific target bug and doesn't kill the rest of the things in the microbiome or the rest of the bacteria that are contributing to health. bacteriophages have been proven effective both in clinical trials but also in field trials in the United States, specifically against Penibacillus larvae. This property of being self-amplifying is an incredibly useful property, and the places where we find the highest densities of bacteriophages, at least in the United States, historically have been in propolis, underneath the soil from healthy beehives, and in other places like that. So beehives that we have today that seem resistant to AFB might in fact already have bacteriophages in them that are protecting them. Finding bacteriophages is simple. So all we really need is soil samples to be sent to scientists. And then through a series of steps, we will basically take those soil samples and isolate only the very smallest particles from those soil samples and apply them to a bacterial lawn and look for evidence of death. So each one of these little tiny spots represents a single bacteriophage that's landed and amplified and killed a huge number of bacteria in that area. That's what we're looking for. Once we find bacteriophages here in New Zealand, we'd like to completely sequence them. We want to test them against a range of isolates or different strains of Penibacillus larvae here in New Zealand. And eventually, we'd like to get to the point where we'll be doing field trials, but this project has just started this month. So today, I'm just telling you about the concept, and I'm asking you to go see my PhD student Danielle and get a sample pack from her. These are addressed and already prepaid envelopes that have sample tools, and all we want from you is a little bit of soil and the location of the hive that the soil came from. And we're gonna start searching for these bacteriophages. And if we find them, in your soil sample, you get to name the bacteriophage. One of the samples that I just showed you was named bee's knees. That's a mycobacteriophage. My students have recently named phages Fancy Pants and Robin. We have Fabio, who's I think an admittedly very handsome phage. So, you know, I have final say on inappropriate names, but we have a little bit of flexibility. All right, so the project is called the ABATE Project. Please see my PhD student Danielle. We're very interested in starting to find bacteriophages that we think might be able to help eradicate AFB in the future. And with that, my time is up. Thanks.